Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. Today I have Svetlana Folimonova. She's an associate professor in the Department of Plantpathology at University of Florida, UFL, and we're going to talk about some of her work. So, Svetlana, thanks for coming.
Thank you very much for inviting me.
Yeah, tell me about your research. What are you focusing on in the plant world?
Well, my background is plant virology, and I'm working essentially with plant viruses. And for a number of several years, I have been working with one of the probably most interesting or exciting plant viruses, and this virus is citrus tristeza virus.
Oh, what is the what kind of plants does the virus affect? Like citrus plants, or is it just the name has citrus in it?
Yes, no. Essentially, the host range of this virus is relatively narrow. The host range of this virus essentially limited to citrus plants and citrus relatives, but there are several varieties of citrus plants, and this virus infects some of them with maybe a little bit different disease outcomes. So in some varieties, there are no essentially disease. In some, the virus could produce very mild symptoms, and in some situations with some varieties and also with some virus isolates, virus can essentially kill and pretty fast one of the trees, for example, some of the infected trees.
So why are you interested in this virus in particular?
Well, first of all, citrus tristeza virus is the most destructive virus of citrus plants. So, for instance, during the last century, this virus killed over 100 million trees worldwide, and it still represents a pretty serious problem, agricultural problem in some regions in the world. For instance, in Asia, in South America, in South Africa. So some endemic virus isolates that are spread in those regions, they're pretty aggressive and they cause quite severe disease that essentially limits citrus production in those areas.
So what's interesting and unique about this virus?
Well, in addition to its agricultural importance, this virus is very interesting from fundamental standpoint. So, for instance, citrus tristeza virus is essentially among RNA viruses, among the largest RNA viruses. So, and if we talk about plant viruses, plant RNA viruses, yes, that this virus essentially is the largest non-segmented virus with essentially non-segmented RNA genome, so among plant viruses. So the size of the genome is about 20,000 nucleotides, so which could be compared actually to some coronaviruses. So for instance, this coronavirus that now everybody is worried about, the genome size of this coronavirus is about 30,000 nucleotides. And our virus, it's a little bit shorter, it's 20,000 nucleotides, but still even 20,000 nucleotides, it's about twice bigger than the average size of an RNA virus, a positive
Does that mean in its capsid or nuclear envelope that the virus is physically bigger, or is it still, in the order of like 50 to 100 nanometers?
So our virus, citrus tristeza virus, has a little bit different shape. It's not, first of all, like many plant viruses, it doesn't have additional membrane or additional envelope. Yes, so it's essentially represented by RNA genome packed into a capsid that is built essentially by two coat proteins, but there are no additional membranes. And the shape of this virus, it's not icosahedral, like many also animal viruses, for instance. So the shape of this virus is represented by a long flexus particle. So the size of this particle, it's like essentially, it's kind of like a flexus rod or flexus thread. So it's quite long. So the length of this particle is about actually 2000 nanometers, but it's very narrow.
Oh, you said it. How long is it?
2000 nanometers, but it's not round, it's not icosahedral, yes? It's just like a long flexus rod. So it's very narrow, but it's long, yes, like a thread.
When you say narrow, how narrow?
I think it's about probably 10 nanometers.
That's super. It's like a needle in a way, you know?
Yes, it's kind of like a needle, yes, but it's flexus.
Does it have any like turgidity? Does it stay? I mean, does it tend to like roll up in a circle, or does it tend to stay loosely straight?
Well, it's not rigid. For example, like tobacco mosaic virus particle, it's like a rigid rod. Yes. No, this citrus tristeza virus, it's kind of like a flexus thread. So it kind of bends a little bit. It's essentially kind of spread still, stretched, I would just say.
Okay. And how does it enter plant cells? I guess with the plant cell wall, I thought usually biting insects, or they'll poke their way into the cells. So how do citrus plants get infected?
So citrus tristeza virus is transmitted by small insect, aphids. So essentially, when aphids feed on the citrus tissue, so they usually feed on the phloem, which transports carbohydrates, yes, throughout the plant. So essentially, aphids, when they feed in the phloem, so for example, if they feed in the phloem of the infected plants, then they pick up a virus. And then when they fly and move to another, like healthy, for example, trees, then they transport the virus to healthy trees. And this is how virus transmission from a tree to tree occurs. So essentially, it's mediated by insects, by aphids.
Is there any ant interaction? Because we're dealing with aphids. I heard ants like to, collect aphids and farm them and, have relationships with them. So do ants come into the picture?
Well, maybe not. I don't think that these interactions would be somewhat specific for the virus transmission.
Yeah, I just didn't know if there's any ants that, like what kind of aphids spread this particular virus? Is it just one kind of aphid or multiple kinds? Do you know much about the aphid side of it?
Yeah, there are different species of aphids, and some of them, for example, Toxoptera citricida, it's very effective vector, so it's very effectively transmits the virus with high efficiency of the transmission. And there are some other aphids like Aphis gossypii, it's much less effective vector. So in those, for example, regions where this Toxoptera aphid is present, yes, then it's quite... So the spread of the virus, if virus gets into those areas, then the spread of the virus in those throughout the area would be very efficient and very quick, essentially.
So I believe you said it was a segmented RNA virus? Like what's the structure of the nucleotides? Or no?
No, it's non-segmented. Yes, so essentially the genome is represented just by one RNA molecule. Some viruses, they may have several segments, yes, for example, from 2 to 7 or even 8, but citrus tristeza virus just have one RNA molecule. It's long, again, it's almost 1,000 nucleotides, but it's just one molecule.
So how is it transmitted? The aphid, it pokes its proboscis into the plant, and, some cell walls get damaged, and then does this virus get injected when the aphid is regurgitating or sucking material up, or what happens? Yes,
exactly. So essentially, aphids, then they have this stylet stylet penetrates inside the cell, and essentially aphids, they feed by essentially sucking this phloem juice, which contains carbohydrates and some other nutritional juices and essentially the acquisition of the virus, yes, and also transmission kind of uploading within another tree or another area occurs during the feeding. And this way, essentially, virus gets into the tree. And then virus essentially, once the virus gets into a plant cell, then it starts uncoating, and then it replicates its genome. It also produces its own proteins, and then new progeny genomes plus all these viral proteins, then they essentially mediate the process of replication, and then new virus particles get assembled, and then virus kind of takes over on its own. It now is able to spread throughout the whole tree. And you probably know that viruses, plant viruses, spread from cell to cell through these channels inside the cell walls, so-called plasmodesmata. And viruses quite often, plant viruses quite often encode specific proteins that are called movement proteins. And these movement proteins essentially mediate this transportation of the virus from initially infected cells to the adjacent neighboring cells through these plasmodesmata. And quite often, virus movement proteins interact with host proteins. Sometimes, for example, what they do, one of the mechanisms is that virus proteins, they enlarge the size, kind of dilate plasmodesmata openings, which allows the virus particle to go through and essentially reach to the neighboring cell. And at some point, then viruses get to the phloem, and also phloem is used as some sort of like highway for the viruses to spread out from initially infected areas in a plant to distant parts of the plant. So our virus actually all the time pretty much stays in the phloem, because aphids insert the virus into the phloem cells, and then virus replicates in these cells, and then it's essentially moves further using, again, phloem cells and is transported to the more distant parts, for example, to young growing tissue.
On electron micrograph, what does it look like? Have you seen, I mean, since these are very long and filamentous,
Yeah.
I would think that part of it sticks in the cell and part of it's hanging outside the cell. How does it get its way all the way in, and does it? And again, does it curl up in a circle, or
No.
does it just hang off the cell, like part of it sticks in and part of it's hanging out?
No, I think that we really don't know, yes, but we would assume that there is some kind of mechanisms that sort of like aligns this, long flexus thread from, say, for example, head to toe. It's kind of aligns at these plasmodesmata openings and allow this long particle to kind of move through.
So you don't have hasn't anyone done electron microscopy to look and catch it in the act, halfway in, halfway out?
Yeah, these images, electron microscopy images, actually were taken long time back, maybe even, in the middle of 60s or 70s. So, yes.
Are they expensive to get done? I mean, are there any recent ones? And maybe you could do some at various points where you can catch some of them at intermediate stages entering, and that might give you some information on their method of action.
Well, I mean, it's known that, and again, people already visualized how the virus particle passes through these plasmodesmata openings. So essentially, the fact that the virus moves as a virion, as a virus particle through these plasmodesmatas, it's known, but we still, the process of how it occurs and exactly what viral proteins and what host proteins interact during this process, it's still not fully understood.
Well, okay, so I guess it answers the cell. Is there a head and a tail, or are both ends serviceable as the head or the tail? What is it? Does it look different on one end versus the other?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yes, actually there is some difference. And a long time back, cluster virus kind of architecture of cluster virus particles got the name of a rattlesnake structure. And why it is called rattlesnake structure? Because, again, the virion is long, but some portion of this virion is probably about 5% of genomic RNA is actually at the 5' end of the genomic RNA. It's covered with so-called minor coat protein. So essentially, two major coat proteins build this virion. Yes, so major coat protein that covers approximately 95% of the genome. Yes, and this minor coat protein that covers this 5% at this 5' end area of the genomic RNA. So it's a little bit different. Yes. So it kind of has a tail, yes, that is built, and not tail, it kind of has a head, yes, that is built by this minor coat protein.
So it enters with its head?
That's actually a hypothesis, yes, that this area, yes, also there are some additional viral proteins are sitting in that area, but they are sitting in some kind of minor quantities. And there is a hypothesis that this end of the virion maybe serves sort of like a motor, yes, that mediates the translocation of this virus particle through its channel.
I mean, if an aphid, I'm picturing like myself to be really small, like this gigantic aphid proboscis pokes its way and, kind of pokes open a few different cells and then stuff comes out of it and stuff goes into it. But I would picture like, the entry is assisted by the cell wall breaking. So, I mean, the virus I would think would have maybe a seeking type of behavior. It would be sensing that now it's adjacent to or kind of in the mix of a cell that's blown open a little bit. And does it go towards the nucleus in the cell? Like, how does it navigate? Does it navigate?
No. This virus, like most probably RNA viruses, it stays in the cytoplasm, yes, because it replicates in cytoplasm, translates its own proteins in the cytoplasm, it gets assembled in the cytoplasm, but also viruses, they are not just, kind of replicating, for example, freely in the cytoplasm. Yes, you probably heard from maybe some other virologists that many viruses are known to produce so-called replication bodies or viral plasm. It's some kind of membrane structures that essentially viruses induce production of additional membrane structures. Sometimes they're called vesicles, yes, and these vesicles, they are used as some kind of sites virus replication, because one of the kind of meaning for virus to replicate inside these vesicles, because virus RNA gets protected from cellular RNases by kind of hiding inside these membrane vesicles, but it doesn't go to nucleus. Yeah.
Well, again, if the cell wall is a hole in it and the virus gets in that way,
yes, exactly.
It's not just gonna sit where it lands. I would think it would migrate to a preferential spot in the cytoplasm, near chloroplasts, near substructures. In human cells, I would guess they would hang out near ribosomes, or there's various organelles in plants too. I would have to think it preferentially goes somewhere once it's in the cytoplasm.
You're correct. So when aphid inserts the virus into the cell, essentially this kind of insertion occurs with the penetration. Yes, with kind of the aphid stylet pokes the cell and the aphid releases the virus inside the cell. And then first step would be for the virus to uncoat, to essentially expose its RNA genome, because citrus tristeza virus is a positive sense RNA virus. This RNA can be immediately translated. And then first RNA-dependent RNA viral RNA polymerase gets translated, gets produced, and then virus starts replicating. It produces additional proteins. And this replication occurs inside these membrane vesicles that the virus essentially induces. The virus induces its formation.
Also, once it packages new virions inside of a cell, again, where do they hang out? These are really long filaments. So I would think they're not going to be straight inside of a cell. They're going to be maybe curled up or in some sort of amalgamation? Do they get tangled? Has anyone looked at that? How do they break out? What does that formation look like?
It's actually quite amazing on the electron micrographs. You can see that virions form very kind of neat and proper arrangements. These arrangements look almost like some kind of crystals as virus particles kind of align, almost align one next to each other in some kind of very neat and proper order. They don't really make some kind of chaotic mess, but quite often they almost aligned, one next to each other. But again, there are some different kind of areas how they accumulate and how they arranged, and it depends on whether they're accumulating immediately next to the point when they got assembled, or it's a little bit, maybe they already start moving towards this, cellular periphery, kind of looking for these plasmodesmata openings. So it could be.
Well, okay, so how do they come out of a plant cell? Has the plant cell healed its wall by this time, so they come out through preferential pores or opening in the cell wall? Or I would think the plant would have to heal the cell wall, otherwise the cytoplasm would just leak out, and then the environment in which the virus would replicate is now ruined and the cell's dead.
Well, first of all, plant cells, they're very protected. It's not just only membrane surrounds. Yes, it's also the cell wall, and even probably some kind of, small hole that is made by the aphid stylet. It may not be actually even, so dramatic. But again, viruses, essentially, once they get into this first cell, so they can also, this virus particle, if it finds the right connection between, to the next cell through plasmodesmata, yes, so it can actually get into the neighboring cell almost immediately. So viruses inside the plant, yes, plant viruses, they travel between cells through these openings in the cell wall, plasmodesmatas. And at some point, then, for example, the virus again could be picked up by another aphid that would be feeding in that cell where virus is, for example, replicating and assembling.
So plasmodesmatas. What do these look like? Are they just round openings in the cells, or what do they look like? And can the virus easily go, I guess, adjacent from cell to cell using these structures? Or
yes, so essentially plasmodesmatas, these are channels or kind of openings in the cell walls, and there is also some kind of membranous system, specifically endoplasmic reticulum, is kind of also forming some kind of inner lining inside this plasmodesmata, and essentially this endoplasmic reticulum in one cell is connected to the endoplasmic reticulum in the neighboring cell. So
yeah, I can see on Google they have like desmotubules that seem to go through these plasmodesmata.
Some cells, we can just say that all cells in the plant are kind of connected to each other.
So it's funny. That's why these viruses are probably shaped like this, so they can do that. If they weren't, they couldn't go through those channels. They would get stopped up and plugged up,
Some viruses, yes, plant viruses, do not have this elongated shape. Yes, they just have kind of classical icosahedral, so they're more kind of round, more symmetrical. So some kind of icosahedral form.
How wide is the aperture of the plasmodesmatas typically?
Well,
I mean, it's big enough to accommodate these guys going through, but
not actually really. So what is actually quite interesting that in most cases, the size of plasmodesmatas is not enough for just a normal plant virus to go through. But it's a typical property that plant viruses do something with these plasmodesmatas. So quite often they have, again, going back to these movement proteins, yes, so they produce these movement proteins that dilate in large plasmodesmatas. In some cases, they also do something with this inner structure of plasmodesmata that you just said, desmotubule. Yes, so the plant viruses some kind of reform this inside structure of plasmodesmata. So they essentially remove this desmotubule, and they quite often put their own filaments or their own tubules that allow the virus to pass through.
I don't understand what you said. What do you mean? The virus will shed some of its structure in order to get through the plasmodesmata? Or, I mean, this one seems like it can go through on its own, but how do they get through the ones that are too big?
Yeah, no, not its own, but it changes the structure of plasmodesmata. So it removes some of the components of the structure inside the plasmodesmata, kind of clears the way for its own for the virus particle to move. So they reconstruct, essentially restructure the inside part of these plasmodesmatas. Because
I think normally the plant cells,
yeah,
the plant cells govern the size of the aperture, and if it also acts like a filter. Mm-hmm. So are you saying that plants govern the aperture and the type of filter that constitutes the plasmodesmata to gate or to control the flow of substances between cells normally?
Yes, I mean plants. There is some process that on the plant side regulates the size of plasmodesmata, and in fact, the size of plasmodesmatas between different cell types and also between different type of tissues is different. But what I'm trying to say that viruses remodel those plasmodesmatas for their own needs.
So viruses cause them to remodel the filter type and the aperture, I guess, of the plasmodesmata. Okay.
Yeah. For example, some viruses remove some of the proteins, cellular proteins that are positioned there by interacting or degrading those. Viruses sometimes form some kind of tubules that they then insert inside of this plasmodesmata. And these tubules are kind of like used as highways, also like a road paves for the virus to go through. But it's quite, it's not probably all very well known, to the very end how this occurs. But for sure, what we know that viruses do many modifications, many remodeling inside the, infected cells. So not only with plasmodesmata, not only with membranous structures. Yes, so viruses remodel inside the organelles. Viruses quite dramatically change the whole transcription profile. So they, for example, upregulate some genes, they downregulate some genes. So once a cell is infected by a virus, it undergoes huge changes, and essentially many organelles already are altered significantly.
So how does this citrus virus cause the death of the plant? Does it cause the cells to blow open, or as it progresses, what does it do to the cells?
With citrus tristeza virus, there are two major diseases. So one is called stem pitting, and stem pitting essentially what virus does. And again, when we talk about virus diseases, yes, and specifically when we talk about diseases that are caused by citrus tristeza virus, we just need to remember that so virus has different variants, yes, different strains, different isolates, different variants, yes. So depending on the virus variant, yes, the disease could be very minimal, or disease could be essentially very severe and could even end to the plant death. And also when we talk about citrus varieties, so not every variety would be similarly affected, similarly susceptible to the virus. So so disease essentially occurs only in certain combination between virus variant and citrus variety. So going back to these two major diseases, so stamp eating occurs in, for example, grapefruits and mandarins and sweet oranges. And what virus does, essentially some of the virus variants, they induce formation of stampedes or grooves. And this production, so essentially what the virus does, and again, alters the differentiation of the vascular tissue. So the xylem phloem, in the tree doesn't produce already as it would normally be produced. So these type of trees show production of some kind of stems or grooves, and essentially they show lack of vigor and they grow pretty poorly, they produce small fruit, and eventually, after some years, these essentially trees can even die. But the reason for such disease, such phenotype, is that virus alters expression of host genes, of plant genes, and it results into altered differentiation, tissue differentiation. So essentially the growth, so the tree already does not really grow normally because it doesn't really produce xylem and phloem in a normal way. So the cambial activity of the tree also is disrupted very much, by invaded virus. And so essentially it greatly affects virus growth.
So what are you trying to figure out in regards to the citrus virus? What's your focus?
Well, essentially, so the main practical goal is to find means how to control this virus. And for instance, when we talk about this stamp-eating disease, there is no current cure against this disease. So, for example, if these virus variants that are capable of inducing stamp-eating are present in the citrus area, so there is really no way, because there is no really natural resistance in sweet oranges or grapefruits against this disease. So there are no really means to control this disease. So the only way to control stamp-eating disease would be to use cross protection or some sort of pre-immunization, so what is cross protection? Essentially, cross protection is an approach when citrus trees get pre-immunized with mild isolates of the virus that do not cause disease, and such pre-immunization of these trees, interestingly enough, protects these trees from becoming infected with aggressive isolates. And
how does this protection occur? Can you say a little bit more about that?
Well, this is very interesting phenomenon, and of course, cross protection could have many different mechanisms, but one of the possible and probably most likely mechanism in case of citrus tristeza virus is the phenomenon so-called superinfection exclusion. And superinfection exclusion is a phenomenon in which a primary virus infection protects the host against secondary infection with the same or closely related virus. And this phenomenon is quite common. So it occurs in bacterial viruses, it occurs in different plant viruses, it occurs in animal and human viruses, including important pathogens of animals and humans and plants. Yes?
It's like a dog guarding a bone. The first dog gets it and it goes, it doesn't like the other dogs get near it, right?
Yeah.
In a way.
Yes, kind of, probably, in a way, yes. So essentially, if virus number one establishes infection in a cell or in a host, then it does something, that makes this cell or plant now becoming unable. So it actually prevents this cell or host from becoming infected with the secondary virus. So, for example, in case of animal viruses, many cases of superinfection exclusion occurs at the level of attachment and entry. So, for example, the first virus somehow downregulates cellular receptors, which essentially make this cell resistant when essentially identical or very similar, very much related virus number 2 trying to get into the same cell. Yes, it cannot already get into the same cell because the cellular receptors been downregulated by the virus number 1. Some viruses, for example, regulate kind of block secondary virus infection at the level of replication. So they prevent or block or downregulate the replication of the secondary virus. So,
although since the aphids, again, poke their way into the virus and, through the wall, I guess once they encounter the cytoplasm, it's a hostile environment somehow. They can't co-opt and do what they need to do because there's another virus already there that stops them somehow.
Kind of, yes. So essentially, because plant cells, they don't have receptors. Plant viruses, they get inside the cells either by transmitting insects or through, for example, mechanical damage. They do not have attachment like in case of animal viruses. Yes, so most of the cases of superinfection exclusion in case of plant viruses, yes, this protection against secondary virus infection would occur already inside the cell. So if, for example, virus number 2 was, for example, brought, like you suggested, by an insect inside the cell, yes, it can be put there by an insect, but it cannot replicate because the first virus would somehow prevent secondary virus from further multiplication. And essentially that would be kind of like the dead end for this virus number 2. It actually, how this protection works, we don't really know, but we have some, to the very end, but we have some ideas, and we know that some of the virus proteins, some of the virus factors actually participate in this process of blockage, the secondary virus multiplication in the same cell.
Interesting. Svetlana, I feel like I've been beating you up with tons of questions. I'm sorry. It's just, this is very new. What's the best way for people to find out more about your research and get in touch? Where can they go?
They can just probably, if they, for example, just Google my name, they will be able, and if they go, for example, to Google Scholar, they will be able to see my publications. Also, I work at the Department of Plantpathology at the University of Florida, so they can also, by just finding this website for the Plantpathology Department, University of Florida, they can find the list of faculty, including me there, and they can just look further for what I do and what kind of publications and what topics, and my publication.
All right. Well, very good, Svetlana. Thank you for coming on the podcast. I appreciate it.
Thank you. Thank you very much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.